From the moment we're born, our DNA dictates much of who we are and who we will become. It's the blueprint of life, passed down from generation to generation. But like any blueprint, it's not always easy to decipher. Misconceptions about DNA abound, leading to confusion and sometimes even fear. Today, we're going to delve into the fascinating world of DNA, separating myth from fact. We'll unravel the complexities of this remarkable molecule, exploring the incredible ways it shapes our lives and the surprising truths that often get overshadowed by popular misconceptions. You've likely heard the statement that humans and chimpanzees share 99% of their DNA. It's a common refrain, often used to highlight our evolutionary kinship with these primates. But is it entirely accurate? The truth is a bit more nuanced. While we share a remarkable amount of genetic material with chimpanzees, the 99% figure is a simplification. Studies suggest the actual similarity is closer to 96%, taking into account various factors like insertions, deletions, and duplications in the DNA sequence. But even that 4% difference translates to millions of variations in our genetic code, leading to the profound distinctions between our species. The world of genetically modified foods is often met with skepticism and concern. A common fear is that consuming these foods will alter our own DNA, somehow transforming us into something we're not. Rest assured, this is simply not how genetics works. When you eat food, your body breaks it down into its basic components: proteins, carbohydrates, fats, regardless of whether it originated from a genetically modified organism or not. These components do not directly interact with or change your DNA sequence. Genetic modification targets specific genes within the plant itself, not the genes of the consumer. Are we merely products of our genes destined to follow a predetermined path? The answer is a resounding no. While our genes lay the foundation, our environment plays a crucial role in shaping who we are. Think of it like this. Your DNA provides the raw materials, but the environment acts as the architect, influencing how those materials are used. From the food we eat to the experiences we encounter, countless environmental factors interact with our genes, impacting our physical, mental, and even behavioral development. This intricate interplay between nature and nurture is what makes each individual unique. Identical twins arising from a single fertilized egg are often assumed to have identical DNA. While their genetic makeup is remarkably similar, it's not a perfect match. Small changes called mutations can occur during development even within the womb. These mutations, though often subtle, can lead to slight variations between the twins' DNA. Furthermore, a field called epigenetics reveals that environmental factors can influence how genes are expressed, further contributing to differences between identical twins. These epigenetic modifications don't alter the underlying DNA sequence but can affect which genes are turned on or off, impacting traits and even disease susceptibility. The rise of genetic testing has led to the belief that our DNA holds the answers to our future health, predicting everything from disease risk to personality traits. While DNA can offer valuable insights, it's crucial to remember that it's not a crystal ball. Many factors beyond genetics influence our health and well-being. Lifestyle choices, environmental exposures, and even chance events all play a role. Genetic tests can reveal predispositions, but they can't predict the future with absolute certainty. Think of it like weather forecasting. We can use sophisticated tools to predict the likelihood of rain, but unexpected shifts in the atmosphere can always alter the outcome. Gene therapy holds tremendous promise for treating genetic diseases, but the notion that it can completely change a person's DNA is a misconception. Current gene therapy approaches focus on targeting specific genes involved in a particular condition. They aim to either replace a faulty gene with a functional one, or introduce a new gene to compensate for the defective one. The scope of gene therapy is not to rewrite the entire human genome, but rather to address specific genetic defects that contribute to disease. It's like fixing a broken component in a complex machine, not rebuilding the entire machine from scratch. For years, the vast stretches of DNA that don't directly code for proteins were dismissed as junk DNA. This term, however, is a misnomer. We now know that these non-coding regions play vital roles in regulating gene expression, influencing when and where genes are turned on or off. They act like a complex control panel, fine-tuning the activity of our genes. Research is continually uncovering the intricate functions of non-coding DNA, revealing its importance in development, evolution, and even disease. Far from being junk, these regions are essential components of our genetic orchestra. In the realm of criminal justice, DNA evidence is often seen as the gold standard, providing irrefutable proof of guilt or innocence. While DNA analysis is a powerful tool, it's not foolproof. Errors can occur during sample collection, handling, or analysis. Contamination can introduce foreign DNA, leading to misleading results. Furthermore, interpreting DNA profiles requires careful consideration of statistical probabilities, which can sometimes be complex and open to interpretation. While DNA evidence can be highly persuasive, it's essential to acknowledge its limitations and potential for error. It's easy to assume that humans, with our complex anatomy and cognitive abilities, must have more DNA than other organisms. However, the sheer quantity of DNA doesn't necessarily correlate with complexity. Some organisms, like certain plants and amphibians, have far more DNA than humans, a phenomenon known as the C-value paradox. This paradox highlights the fact that genome size doesn't always reflect the number of genes or the complexity of an organism. The human genome, while vast, is actually quite streamlined compared to some of our fellow inhabitants on Earth. The C-value paradox challenges the intuitive notion that bigger or more complex organisms should have larger genomes. This paradox arises from the observation that some seemingly simple organisms possess far more DNA than complex ones. The explanation lies in the fact that a significant portion of an organism's DNA may be non-coding, serving regulatory functions or representing remnants of ancient genetic elements. The amount of coding DNA, the genes that directly translate into proteins, is often a more accurate indicator of an organism's complexity than the overall size of its genome. The C-value paradox reminds us that the relationship between DNA quantity and biological complexity is more nuanced than meets the eye. Our DNA is a testament to the interconnectedness of life, a story written in the language of molecules passed down through countless generations. It holds the secrets to our past, present, and future, shaping who we are and influencing the trajectory of our lives. But like any great scientific endeavor, the study of DNA is a journey of continuous discovery. As we delve deeper into the intricacies of this remarkable molecule, we uncover new truths, dispel old myths, and gain a deeper appreciation for the profound impact it has on our world and our understanding of life itself.